Are you ready to get started, Brian?
Sure.
Okay, for everybody online, thank you so much for joining us today at Hatfield Marine Science Center's afternoon talk. Boy, my brain just stopped working there. My name is Cinnamon Moffett, and I'm the Research Program Manager at Oregon State University's Hatfield Marine Science Center, and I'm going to attempt to be your host today, so thank you so much for being with us. If you haven't been with us on one of these seminars, just know that we do have your mics, cameras, and screen shares disabled for this event, but we would love for you to put in any questions you might have. Just use the chat function on Zoom, and we will get to those at the end of today's presentation, but feel free to put them in the chat at any time. I wanted to let folks know that we are also recording this event, so if you missed something or you would like to share it, you will be able to do that by going to our past seminar page in a few days. And I'm going to put that link in the chat right now so that you have it, and we'll be able to share this recording of today's event. I also wanted to just do a quick little promo for what's happening in the next couple of weeks. We are not going to have a seminar next week due to the long holiday weekend, but I'm hoping that folks will come back and join us on July 8th when we have Dylan Gomes from NOAA's Northwest Fisheries Science Center, who will be here to talk to us about how animals respond to natural noise. So I'm hoping you'll come back and join us again, basically same time, same place, but on July 8th for that presentation. If you want to know anything more about any of our upcoming events, you can just Google or use whatever platform you use to go to HMSC's homepage. Scroll to the bottom of that homepage and you'll see a calendar of events, and all the login and link information for all of the events that we have at Hatfield are located there. Also, just going to put in a little request. If you have anybody that you would think would be good at having a seminar and sharing with the Hatfield community, we're still booking Zoom remote seminars through the end of September, and I have a few open dates. So if you have somebody you would like to have share with Hatfield community, please reach out to me and we'll get them on the calendar. For today, we are happy to host Brian Wells, the fisheries biologist with NOAA's Southwest Fisheries Science Center, stationed here at Hatfield. His work is based on spatial temporal variability in ecosystem dynamics linked to environmental drivers. And so we're excited to learn a little bit more about the work that he's doing. And so Brian, I'm going to hand it just directly right off to you and let you take it away. Thank you so much for being here.
Thanks. Okay, looks like my mute's off. Thanks guys for joining in. As you can see by the title, Process Studies to Quantify Ecosystem Dynamics in the California Current. We're going to focus on Central California, but before you think that's rather myopic, it happens to be a fulcrum along the entire California Current. It's one of the larger forage bases, larger bird colonies, larger areas of marine mammals. So it's a critical location along the California Current and really well suited for the type of work we're going to discuss. Now, the overarching objective is to quantify physical and trophic interactions in the ocean affecting salmon dynamics to provide support for improving management of salmon. And what we're going to be doing basically is I'm going to walk through, I'm not going to fill each one of these boxes in as we go, but this gives you an impression of the various aspects that we're going to quantify and we're going to understand mechanically. We're going to start in that upper left corner of the physics at the largest scale, working our way all the way down to the bottom right corner with that red box predation. And in between are all what we believe to be the processes that drive Chinook salmon. Now, to do this, we have to take an approach which is starting to really gain steam, and that's ecosystem oceanographic approach. And to understand what ecosystem oceanography is, you need to put it in the context of what it's not. On the left here, this comes from Curry et al. 2008. On the left, you can see, for example, I'm showing you 3 different critters from that paper. And fisheries would be about understanding their distribution, their growth, their mortality, their demographics. Fisheries oceanography, like the middle panel has, it's taking those things. In this case, you have the 3 colors indicating the 3 different recruitment dynamics of those critters, and then connecting them in some bivariate or multivariate method to the environment. And in doing that, we're really just treating them as one thing drives another thing, oceanography driving the fisheries dynamics. Ecosystem oceanography is a bit different. What ecosystem oceanography does is it looks at the interrelationships. It looks at the trophic dynamics. It looks at the drivers, looks at the interactions, and then importantly, looks at the sensitivities of each one of the components to one another and how those sensitivities, arrows, colors, and diagrams change under different environmental and human impacts. And we have to take an approach like that, this ecosystem oceanographic approach, ultimately, as you'll see, to understand salmon dynamics. Now, from the ground up, you can't just go out and sample salmon in the water, and you can't go sample forage in the water and the birds in the water. You have to think of your sampling on a boat or in any way you're doing it as one synoptic event. Where are the salmon, where are the forage, where are the predators, and put them together to understand the process and why, when, and where they are interacting. So that's the biological data. But we can only be out there for a snapshot. We're only out there for the month of May or what have you. So it's important to not only have the environmental data from such things as CTDs, but to bring to the table things like the acoustics shown in the bottom right, where you can actually start discussing a spatial arrangement of the forage beyond the spatial arrangement of our simple samples. So you can get krill and a variety of other things. And then to fill in those gaps in between when we're actually out there observing things, we can use remote sensing. Or what's really coming to fore, very importantly these days, is biophysical modeling. It allows us to really fill in the spatial dynamics and the temporal dynamics when we can't see what we're doing. We'll also see that by coupling our observations and our processes determined from hypothesis testing to these biophysical models, we can actually confirm our beliefs as well as start asking questions out of the scope that we've actually observed. And it gets very exciting, and I think you'll like seeing some of that. Now, I'm going to, believe it or not, tell the story about the Central California ocean ecosystem by only looking at 5 critters. Now, these 5 critters tell a lot of stories within themselves. They're sentinel in their role. Each one represents a whole bevy of things that can occur around it. For example, 2 of the most prominent forage items in this system include juvenile rockfish and krill, born the year that salmon are coming out and the year that we'll talk about. They're born in the actual year of the oceanographic conditions we're speaking of. And juvenile rockfish and krill in our system, and a little different than in the north, but they are very common when we have a lot of upwelling, cool waters, what we would often refer to as productive systems. What's nice is we have an anti-corollary to these juvenile rockfish and krill. In fact, when it's warm and upwelling's not very good, and we would argue it's not productive, anchovy from previous year classes wash into the shore. So basically, we get 2 different prayscapes, and they're rather dynamic. That is the krill and rockfish prayscape and an anchovy prayscape. And reacting to this prayscape are a number of predators, including a very significant one in our area, and you'll see that as we go along, the common murre. The common murre switches between these 2 prayscapes and changes its behavior between these 2 prayscapes, and therefore reacts to changes in environment as represented by the change in the prayscape and the distribution of prey and the like. And finally, because we're talking about salmon at some point, salmon enter this ecosystem naively. They've spent all their time in fresh water. While some correlations do exist, they've largely been ignorant of what's going on in the ocean. And then they come in May, and they meet whatever ocean system you have, whether it be rockfish and krill or anchovy, and they meet predators like murre, and you'll get to see the synergies that can occur during that interaction. We'll break it into simply basin scale influences and preconditioning, setting our system up from afar. We'll look at the spatial and temporal ecosystem dynamics locally, the mesoscale orientations. And then we'll look quickly at a way of bringing all these relationships together into one structure where we can actually look at the trophic relationships and look at how the environment is driving them. And only then, after we actually set up our ecosystem, will we put salmon into it and ask the question, what will happen to salmon when they come in contact with it? So let's start at the broadest scale. And that question was set up over a decade ago. If the California current is stronger as driven by, let's say, North Pacific high, does that affect salmon? In fact, it did. We find that as North Pacific high increased and the California current increased, along the California current, we saw all populations of salmon as indicated by these up arrows. We don't need to worry about the individual populations. All of them grew more, and later were confirmed they survived more. So right off the bat, we could stop there to some degree from fisheries oceanographic standpoint and say there's these big drivers that affect salmon survival. But that's as broadly spread as you can get. If you were to rely on just faster current, more survival, that's going to ultimately fall apart due to the lack of process understanding. So what we want to do is we want to dig our way from that giant basin scale driver to the salmon response, one step at a time, all the way quite literally to the salmon's mouth. And the first step was, why does the North Pacific high matter? How does it play out? So what I'm showing you here in the upper figure is colored circles. And each one of these colored circles represents the average location of the North Pacific high in the 12 months of the year over about 30 years of observation. The color of that circle indicates the size of that North Pacific high, red being bigger. Basically, you get a waxing and waning of it going farther offshore, but much larger in the spring and summer. And what's interesting is that little inset figure from March shows the average or the actual location of the North Pacific high across all of those different years and its variability in strength, weak years, strong years. And our question was simple. If, say, the North Pacific high and the strength of the current matters, how does it play out? Well, it turns out that the location and strength of the North Pacific high in March can tell you a lot about what you can expect to see in the coastal ocean system in the spring and the summer. For example, if I come down here on the x-axis, this is a basic metric on how close it is and how big it is to the coast. We see, in fact, that the closer and bigger it is on the coast, which roughly is what that number is, the greater the productivity is on the y-axis that we observe on the coast in the spring and summer. For example, in this particular case, seabirds produce more offspring, juvenile rockfish are produced in greater numbers, we get more krill, a variety of things. To repeat, the location and strength of the North Pacific high in March, a period of time typically associated with initiation of upwelling in the California current, you get more productivity on the coast when we go out in spring and summer, the closer and stronger that North Pacific high is in the late winter in March. So we're starting to walk from why does it matter to how does it matter. But that still doesn't get us toward any mechanism, any real true understanding of how does this play out. But we can look more locally and tell a story. This top figure is our observed krill in the black and our observed rockfish in the gray time series. We're out in a boat in June, throwing a net in, and we're counting our krill, counting our rockfish. So this is the observed variability in the system. If you look down here at the bottom left map, what we've done is we've gone back to March. We've taken this krill time series, this black time series, and correlated variability in that time series using ROMS models to upwelling strength at each one of these 10-kilometer resolution spots. In other words, does this black line correlate to upwelling at any and how many of each one of these spots? What we find is that it does correlate very strongly in March. In fact, March is the one that has the most structure and the greatest correlation to that line when you go through all the months. And it's shaped exactly like an upwelling plume. Ultimately, what this says is variability in krill on the shelf in summer is correlated to strongly upwelling dynamics locally in March. Now that can be easily attributed to the introduction of nutrients. And if you follow the work of Don Kroll and a few others, it takes about 100 days to go from nutrient introduction to observable large krill in a net. So that's very confirmational. This is how the North Pacific high effectively creates krill variability on the shelf in summer. Now I want to look at rockfish in the gray, that other very important prey associated with upwelling dynamics. When we correlate that gray time series of observed rockfish in summer to the same ROMS upwelling values for each one of these black dots in March, we get this fancy horseshoe pattern. Understanding a little bit about the biology of rockfish makes this understandable. Rockfish are born, released February, March on the shelf break, right there at that horseshoe. They come up with a yolk sac, so they're not immediately looking for food, so it's not a critical period type of issue. In fact, we argued that it's about the right kind of transport to trap some of those juveniles right where we're sampling, which are these black dots actually in the summer, right where we're sampling. In other words, our argument was upwelling in March in this pattern correlates the variability in the rockfish in spring and summer due to transport and retention in the area. That wasn't quite enough to confirm our ideas. So we went to otolith chemistry. What we've done here, is this figure here shows barium in the water column at the surface during upwelling. Barium is richer in fresh water where it hasn't been nutrient depleted, so barium is very strong in upwelling conditions. Hence this bright red in our water. We then took otoliths from a lot of juvenile rockfish we collected on the shelf, keeping in mind that horseshoe pattern's right out here in the upwelling. We collected otoliths from juvenile rockfish on the shelf, and we looked at the last week, say, of barium values on those otoliths. What we see is this big shift to barium in the otoliths is somewhat south and trapped on the shelf. And when we do a correlative analysis on a polar plot, we actually see exactly that. Barium is great in the otoliths, right at the upwelling plume, and then smeared south of it. So in other words, the March North Pacific High, creating local upwelling characteristics in March and transport characteristics, ultimately can transport these salmon onto the shelf where they sit and await for our capture in the spring and summer. Those same dynamics for krill are really just about bringing the right amount of nutrients in due to that March upwelling. Now, going back to this idea that you get two kinds of prey scapes. We've been talking about krill and rockfish in spring and summer when we go out there. And in fact, if we look at, now referring to them as subarctic conditions, cold water coming from the north coming into the area, we find that, as shown here in these two figures here, the amount of krill we get in this seascape and the amount of rockfish we get in this seascape is negatively correlated to the temperature. It's very strongly negatively correlated to temperature, confirming this idea that during high upwelling conditions, cold water, you get more rockfish and krill. But to further confirm this idea of an anti-correlated or other prey scape is anchovy. Specifically, when those conditions get warm and water from offshore laps inshore, it can bring last year's and previous year's anchovy onto the shore, creating that prey scape, an anchovy prey scape. And that can be seen by this positive correlation with temperature. So what I've said to this point, the strength and size of North Pacific High in March creates upwelling and transport dynamics on the shore that ultimately result in nutrient introduction, appropriate transport to accommodate krill production in June, krill to be available in June and the summer, and transport that makes rockfish available through the summer. And anchovy when those conditions are not conducive. But the spatial arrangement on the shelf is also of great importance, and we'll see that play out when we talk about predation dynamics. What we find in this area is, in fact, when you get that cool subarctic condition, you get rockfish on pretty much all of these green areas from D north. You get krill in most of them. But during those warm, poor onshore flows, you get anchovy, shown here in the orange, largely right along the coast in these C, D, and E. So when you get anchovy under poor conditions, it's tightly on the coast and southern. When you get rockfish and krill during good conditions, it's largely distributed across the whole system. Now, what I've shown is a series of regressions, basically. This leads to that, leads to this, leads to that. And that's interesting, but it's not a holistic way of looking at it. It's not a way of evaluating the sensitivities. A way to do that would be something akin to a model of intermediate complexity, in this case represented as a structural equation model. And all I've done is I've grayed out anything we haven't talked about. What we have talked about is that transition to upwelling, how strong that upwelling is, krill production, offshore transport, and rockfish. And this structural equation model was built such these arrows, blue being positive relationships, red being negative relationships, and the size of each representing the importance of those relationships, confirms everything we just said in one structure. Namely, you get more rockfish when you get more krill on which they eat and are also correlated. You get more rockfish when offshore transport is less. Not that nice horseshoe trap that can occur. You get more krill when you have positive upwelling, which just happens to be negatively related to sea surface temperature, that's why that's red. And the later the transition, the few krill you get. Without getting the specifics of this, really the point is, We can take that whole story and put it into one framework, which allows us, in this case, to look at the sensitivities to each one of those components. So that's the setup of the ecosystem. To reiterate and kind of just bring it together, because I know I've given a lot of information. Spring regional ecosystem productivity is set up afar during late winter by strength, location, and size of the North Pacific high, and the coastal nutrient introduction and transports associated with it. Namely, if the North Pacific high is strong and close in March, you get nutrient introduction for krill and appropriate trapping for rockfish on the shelf. The mesoscale physics and resultant community spatial temporal dynamics indicate a dynamic shelf ecosystem. So where you get krill and rockfish is different, more widely spread, than where you get anchovy during the less productive conditions, which will become very important in a moment. And finally, these bivariate regressive ways of looking at things should be stepped back and put into the framework of a more holistic view, such as a structured equation model, ecopath, ecosim, Atlantis, name it. I've given one example. The beautiful part about that example is it's confirmational. When we do the analysis, it supports what we believe the individual relationships were. But by putting them all together, you can actually understand the individual sensitivities to each component, which is critical when it comes time to ask questions about current and future state. But this is a story about salmon. Remember salmon? What happens when they go to the ocean? They're going to come out of the ocean. It's going to be rockfish and krill, or it's going to be anchovy. It's going to be great upland. They are a wonderful test species because they are ignorant of what has happened, and now they're going to have to deal with it. So the first question we might ask is, if any of this is important, there should be some relationship to when the salmon go to sea and how well they survive relative to matching the prey that are at sea. What we've done here is we've taken a number of coded wire tag release groups from the Sacramento Valley. We've looked at the survival of each one of those release groups by day, lagged from the time when upwelling began. In other words, this 100 and that peak says that your likelihood of survival is greater if you come out as a salmon approximately 100 days after upwelling. Well, that goes right back to our understanding of those March dynamics. That's how long it takes for krill to grow and the forage base to develop. So this is really nice. What this argues is there's best times to go out, and those best times relate to the same kind of setup dynamics that we've been discussing. But this doesn't really confirm that. This doesn't look at the prey itself, but we can also do that. We're out in a boat down there and we're collecting forage. And that's what this x-axis is. And we find the more forage in the system in summer, these are rockfish, krill, a variety of things, the fuller the salmon are. And you get that nice relationship here. And basically, this confirms to some degree that if you have that setup, if we discussed it in March, you get more forage, and the salmon that come out at the right time will be more full. This 13 is an interesting year, and we'll see that play out far down the line. This is a year of intense upwelling, very intense upwelling, and it blew the forage off the coast. So there's an optimal window we need to think about. The other critical part of this presentation, or this paper, was we learned that not only is fullness related to the amount of forage in the area, but it's that distribution of the forage. It's about having frontal dynamics, about having plumes one can eat at. It's about accessibility to the increased or decreased amount of forage that's at sea. So those March dynamics set up the forage, salmon that come out at the right time, feed on that forage. The more that's there and the better accessible it is, the fuller they are. So what are they eating and what matters? Largely, even at their largest size in the first year, it's 60% krill. So if we look at krill, we learn a lot. Specifically, as the salmon come out, they go to this area B. And area B, this is that black line I showed you before, is highly variable. But what's nice is the more of that diet that is krill, the fatter those salmon are. So in fact, now we're seeing that real connection to March nutrient dynamics, bringing us krill, salmon matching that krill, getting fuller because there's more krill, and if it is krill, a fatter condition. So we're starting to really dig in down the line of that large figure of boxes. We're working our way to the right. We're seeing what's making salmon fat, and now the ecosystem is set up to make them so. But this is the beautiful part, is now we have a story that we need to test. We need to have some confirmation of our models. We need to understand the spatial temporal dynamics in a way that we previously have not been able to. What we've done is we've matched this to an agent-based approach where salmon particles are released at sea. They are allowed, the only rules we have are go to the higher concentration in 5 boxes to the higher concentration of krill, with a little randomness. And when they get to that krill, eat that krill, and given the amount of krill they're able to eat and the temperature associated with it, using a dynamic energy budget model, we can grow these fish. So it's a really simple set of rules that the salmon particles are under. And we find, in fact, that if we take a number of good survival years from other studies pulled out of the literature, and we look at those good survival years, we see that salmon particles distribute largely in the whole area, and that red indicates they grow very well. So it goes back to this idea that they go to sea and they grow well if it's a good survival year. During years of poor survival, we start seeing a compression. They start growing right along the shore. The best growth, in fact, is found right here in these areas in the green, just somewhat south. So this is conformational to some degree. Good years, the model with simple rules is able to capture good growth, good survival, bad growth, bad survival. But we can take an extra step and actually look at the dynamics of the ocean while we weren't out there as well. For example, these 3 figures, the top one being temperature, then phytoplankton the second, zooplankton the third. The x-axes are simply the month of the year, and we've plotted the temperature in the top figure for years of good survival in the blue and bad survival in the orange. Very happily, the temperature drops dramatically in those years of good survival right at March when those upwelling dynamics kick in. Similarly, we see this phytoplankton boom at that same time on the heels of that increased upwelling or reduction in temperature. And what's very happily is you get that krill peaking right around June, July, after that upwelling dynamic begins. So this is all wonderfully conformational. And to some degree, it's also pretty useful because now we believe we have a model that we can build on, and we are building on it, to actually start asking questions from a managerial perspective. And we'll see that as we move along. But really what's cool about this model, what I'm showing you in that top figure is the survival of salmon from 30 years, I'm guessing up there. I'm sorry if I can't remember correctly. But that's the estimated survival using coded wire tags. This is the estimated survival on the y-axis of using the spatial arrangement of growth. What we find is you can very closely match survival of salmon by simply looking at that growth at sea. Now alone, this model alone is kind of cool and neat, but really having those years of process study attached to this model provide a true ecosystem analysis and modeling format to understand the mechanics of what's going on and the ramifications of variability in those mechanics. To show you how well we think it fits, the bottom figure is the same data, just shown as time series. The coded wire tag, coded wire tag is in blue, and the model results looking at spatial arrangement of growth is in red. So once again, we feel very comfortable about this. So that's very conformational. We believe we see processes. Winter dynamics, upwelling, creates the right nutrients to bring krill, right transport to bring rockfish or anchovy if it's the opposite side of the coin. When salmon come out about 100 days after that upwelling, they intercept krill at the plume area as they soon come out. They grow fat. When they don't grow well, they end up dying. You can fit this to a biophysical model. To really stop here would be very exciting. But we're not done yet. Why are they dying? Why are the small ones dying? We can test this by looking at the size selective mortality. This is a lot easier than it's going to sound. So I'm sorry that it might sound more complicated than it is, but I assure you it's not. We sample the same population of juvenile salmon 3 times. As they come out, we sample them in May and June. We sample them in the larger area because they spread out a little bit in July, and we sample them again in the fall. We did this for 2000 and 2005. So 3 sampling periods of the same population in each of these 2 years. 2000 happened to be a boom year for survival and extremely productive. Rockfish and krill everywhere. More than you need. 2005 was quite the opposite. Anchovy everywhere, very little krill, very subtropical conditions. So our question was simply, do we see size selective mortality on either or both of these years? One of good growth, one of less growth. What we do when we go out and we sample this population each of these times, we pull out the otolith and we grind that otolith as shown here at the bottom. And on that otolith is an emigration check. So we know for an individual fish, by that yellow line correlates highly to the size of the fish, generally speaking, how big the fish was when it went to sea. And we can look at the distribution in May and June when we sampled them, how big they were when they went to sea. We sampled them again in the summer, and we retrospectively from just that part of this, just that period of time, estimate what do we think the size of emigration was from these entry checks. And we look at that distribution. We do it again for the fall. If every time we revisit this population and we estimate the size of emigration using the otolith, the distribution doesn't change, then there was no size selective mortality. It didn't matter how much you grew, as long as you grew enough to live through generally. And in 2000, what we saw was, when we look at the distribution of those emigration sizes shown here in the white bars, they spread across from about 75 millimeter emigration size to about 110, with a little bit of a bell. In summer and fall, we go out and do the same thing. They overlay each other perfectly. 2005, however, when we went out in the summer, we see that same distribution basically of emigration size in the white, but in the spring, I'm sorry. By the time we went out in the summer, we estimated this emigration size. It had shifted by a dramatic 10 to 20 millimeters to the right. Now, what this indicated clearly was during these years of bad growth or bad oceanographic conditions, they actually experienced greater size selective mortality. So it seems then size always matters pretty much, but from a population during bad years, small guys are called right out. So why would that be? And we argue simply it must be predation. And now we bring the murder to the table. We ask the question, well, how do they feed during good years? How do they feed during less productive years? And how could this possibly affect salmon? That top figure shows you the concern right off the bat. That's the number of common murre on the Farallon Islands from 83, in this case to 2011. Here's the Farallon Islands, that little star right there. These salmon come out, enter B. These common murre feed off of the Farallon Islands area. Now, when conditions are productive, in other words, when March conditions were, there was a lot of upwelling in March, you had transport, nutrient dynamics that brought krill and rockfish to the area. Common murre would feed by going out finding fish and bring them back to their young at the island here in the dock. And you see that there's loose aggregations. They're feeding largely everywhere. Reminding ourselves that everywhere there are also rockfish and krill. In this case, it's only worried about rockfish, we'll see. But they're pretty much everywhere, and hence the birds feed kind of everywhere. But during these low production years, the birds either fall right off the island and eat as best they can, or they come a little farther south to hide across the shore here. Reminding ourselves that during low production years, there's an increase in anchovy, and they tend to be down here tight and in the south. And in fact, when you look at the diet of these common murre, shown here as time series in the top figure, we see during rockfish, productive conditions in the blue, they can eat as much as 90% rockfish. And they switch pretty dramatically between rockfish and anchovy. If there's no rockfish, they eat anchovy as much as 90%. So flipping between these two, depending on the prey scape that they're interacting with. What we find, in fact, is that there's no salmon there hardly. So why are we talking about this? Well, this goes back to that understanding process. It goes back to that understanding the interactions, not treating everything as a regression between A and B, but in fact, A and B are couched within C, D, E, and F, and what have you. We find when we took a closer look, we actually see in this figure at the bottom here, more anchovy during those anchovy conditions these birds are eating, the more salmon they intersect along with those anchovy. Now, there are about 400 anchovy for every salmon during these years, if you look at the net data. And yes, it's only 10%. But when you consider the 250,000 birds that feed multiple times a day for a long time period during the summer, this actually on the back of the envelope turns into dramatic and significant effects on the survival of salmon during these poor conditions under which anchovy are the predominant prey. And we can see this by cobbling this entire presentation into this one GAM. And what this GAM does is it breaks different environmental and biological variables up and runs survival of salmon in each one. Now we have to talk about freshwater, obviously. And that's what this top figure simply says. And this is going to come back later as well. And it simply says, these are anomalies, below average, above average. The better the April discharge is during that time period these salmon are beginning the migration to sea, the greater their survival of the salmon from coded wire tag analysis. So in fact, that's important. You want them to be healthy, fat when they first enter the ocean. That's a critical part of their survival. But going back to what this study has talked about, or this story we're talking about, is it should relate in some way to March upwelling. So survival of salmon from coded wire tags, 30 years of data, and March upwelling from below average to above average. We see this nice optimal window. We see that you need upwelling in March to improve the survival of salmon, but too much. Remember that 2013 year where there was very little forage in the diet of salmon. That 2013 is way out here in the high upwelling period. But this is very confirmational that March dynamic matters. And what's really cool in this figure is the synergies that can occur. This is an isopleth. Red is low survival. Blue is high survival of salmon. We've got on the x-axis the average amount of juvenile rockfish associated with those productive conditions you see in the nets of our ships when we go out in June and July. On the y-axis, we see the number of common murre on the Faroe Islands. And we see if we forget common murre completely, and just look all the way down here at the bottom, 10 common murre is well below, it's at 0. We see that survival of the salmon, regardless of the amount of rockfish you have, is pretty much from median white to a little bit above average in the blue. It's only when you get variability or reductions in the rockfish, along with those increases in common murre, that you start seeing this dramatic drop in the survival. And this is the longest way in the world of saying, when you don't get rockfish, they eat anchovy and eat salmon. This is a direct result of nutrient and transport dynamics as early as March, driven by basin scale factors like the North Pacific High. But now when I say we, I speak loosely, it's Kelly Vasbinder and Jerome Fichter at UCSC. We're taking the models to the next level. Previously, we showed you over here, good survival growth, bad survival growth. Well, you can actually take the average growth just continuously during all the years of data and plot them. These are the individual years of salmon growth across each year for which the model was run. The average is here in the black. So you see quite a bit of variability in that growth dynamic of salmon from the time they enter in May till approximately October, November, I believe. What's interesting is if we go to a recent diet database that we're developing in collaboration with Joe Bizzarro, we can start asking the question, what is the size distribution that common murre are doing or eating? And we can use that size distribution, the mean and a standard deviation, let's say. We can overlay it on these growth dynamics as modeled by the biophysical modeling effort that we showed earlier. And we see, in fact, during poor years of growth, fast years of growth, you can come out of that gate largely by mid-June. But during those poor years, limited upwelling, lack of rockfish, lack of krill, these salmon come out, murre needs something to eat, they eat anchovy, they intersect salmon. Well, along with those poor years, not only do you get eaten by murre, more likely, it can take 2 months before you actually can grow out of the murre gate. So it's a double whammy. And this is where we see the effect of growth, predation, tied in with environmental drivers and variability in the prey scape. And that's how we fill out this. We started up here and went straight all the way down to here. Here. And that's interesting, but it's really all of the stuff inside that we just sat here and talked about. That's really interesting to me. It's understanding the timing and the strength of upline dynamics, the resultant primary productivity, variability and distribution of forage associated with it, and then the intersection of salmon as they come out with that ecosystem, and the effects of predation on them. And all of that's really fun. That's what we have done over the last decade and a half. But we are stuck with this idea of, so what if one can't use it? So what we did was we started working with another group that was doing a life cycle model. And we asked, here's the information we can bring to the table. And with a life cycle model, inherently, you've got the various things with which the salmon interacts through its life cycle, and you can figure out the sensitivities that salmon return numbers have, and then ultimately from that, start really figuring out what levers you might need to pull to make the salmon react whatever you want. More salmon, less variability, your call. So this life cycle model was created by Whitney Friedman et al., with some of our help involved. And it simply starts up here about 11 o'clock. The fish come back, salmon come back, and goes around. They have their eggs. They migrate out of the river after they grow up. They enter the ocean, intersect with productivity and predation and the like. And then they have multiple years of aging and then return and lather, rinse, repeat the next cohort. This model was extraordinary. Over here, you see in these black bars, the actual observed return numbers of Chinook salmon to the system. In other words, the number of salmon right here returning. The blue bars represent the capacity of this model to meet year to year and variability in those return numbers. With an R-square of nearly 70%, one could argue this model is pretty awesome. But what's really neat about it is when you actually do AIC or any version of cross-validation and figure out what the salmon are most reacted to, most sensitive to, you find three variables only, really. You find temperature when the eggs are on the nest. You find the amount of flow in April, as it turns out, like we showed previously. And you find the amount of predation at sea, in this case, indexed by MER. We got to remind ourselves, MER aren't the only predator, but they do seem to track general predation. So these were the only three variables that ended up driving that match. There are sensitive variables. Flow can be regulated, for example. What's interesting down here, though, if you look at these gray bars, they're the relative contribution of each of those variables to the total model. Temperature occurs very early on when mortality is incredibly high on eggs anyway. So while important, ultimately its impact through the life cycle seems to be relatively low. Doesn't mean don't pay attention to it, but it's not super sensitive. But among all these variables, it is one of the ones it is sensitive to and can be managed in some systems. For example, those that have dams associated with them. Flow, same thing. That can be managed to some degree. Better flow can provide greater return numbers, so to speak. What's interesting here is marine predation is the strongest contributor to the model. And understanding the processes we've laid out here, from North Pacific high in March through to the introduction of rockfish and krill, the intersection of salmon with those rockfish and krill, its increased growth and distribution and ultimately survival, and the likelihood during poor conditions that intersects a MER, there's a lot going into simply writing the word marine predation. There's 15 years of work going into writing the word marine predation. But it does give us some insight. That is the driver of all the drivers we put in that seems to be causing variability. So this goes straight to best management options. And those management options are going to be in fresh water unless you're willing to do some drastic measures at sea. So increasing or doing the right flow, doing the right temperature can mitigate, theoretically. And we actually ran those strategies, and it did, in fact, mitigate a large part of variability associated with marine predation. So I'll wrap it up here. What does it all mean to salmon? We talked about the setup of the ecosystem, how it got to what it was, and then we brought salmon to it. Well, abundance, timing, and location of prey are critical to salmon survival. More krill, they get fatter. They have to come out at the right time for that krill to be there. During subtropical conditions, there's an anchovy, or I write poor, I guess that's a value judgment. A poor prey scape and slow growth of salmon leads to increased predation. And what's interesting is going to a modeling framework, we can actually see that that slow growth can place them in the gapes or preference of one of the most dominant predators in the area for an extended period of time, creating a double whammy. And to be a little accountable for the fun we've had, we can work with assessment biologists to actually try and bring this process understanding to a framework where we can actually identify sensitivities, potential levers, and then test those strategies out on the bed of a real understanding of what's going on. And that is how we pitch this idea of bringing an ecosystem oceanographic viewpoint to the table and ultimately transitioning it to a managerial application. With that, I really appreciate your time. I know there's a lot of information there. I managed to keep on time somehow though, so I appreciate it. Thank you.
You did wonderful. Thank you so much. For folks online, if you have any questions at all, you're getting claps, by the way, virtual claps. I know you can't hear them, but if anybody has questions online, go ahead and type them into our chat box and we can work through them there. Brian, I had a question. Some of the slides that you showed had different end dates. So some of them, it looked like you were looking at a time block that stopped like 2010. Some of them went up to like 2015. Did you do any work where you were parsing out like what the system is doing in these last few years where we had the heat wave and that kind of stuff versus prior, and how that might change the model as we continue to move forward?
Interestingly, two things. There's different year periods because studies were done in different years. This all sounded very linear, but if you actually looked at the dates, we'd have had to go back in time for a lot of these things. Interestingly, when we actually look at these heat wave components, there's someone online that can answer this question far better than I, but I'll make my crack at it. We actually confirm a lot of these results. When we talked about common murre coming inshore during those poor conditions, or aggregating inshore into those poor conditions, we see the same kind of thing when the heat wave came. We get a habitat compression. And the same effect we saw in salmon ultimately affected the interaction of marine mammals with crab pots. And there's associated fishing issues like how long the crab pots were out there and so forth. But we actually used the understanding I just laid down to build the processes we saw when the heat waves came. So we actually had more of a confirmation than a rejection of what we see. Now, the place where rejection is going to be very easy to see from what I've said is right now we have a lot of anchovy coming into our system. There may be so many anchovy, in fact, that a lot of the mechanisms I'm defining specific to common murre's effect on salmon might be somewhat reduced.
Yeah, that was kind of my follow-up question is, what are you seeing this year?
More anchovy.
More anchovy.
Which is good for salmon because that means they may be able to buck the system when the system goes to crud.
Interesting. It's a lot of work that you presented to us, and I appreciate your time. Yi-Chang, do you want to come on the line just real quick and promote what's happening in a little bit, if you are around?
Yeah, just want to let everyone know, particularly our students, we have a number of summer interns. We've also invited our graduate students, summer session students, and postdocs to join us following this, following the seminar at 4:45 to 5:15 for an informal chat and discussion with our speaker to learn a little bit more about Brian's pathway, career pathway.
Thank you, Yi-Chang, and thank you, Brian, for being willing to speak with our students. We have a lot of folks that are either on site or virtually in the world somewhere that are participating in internships with us, and it's nice for them to have an opportunity to talk with you. So I'm not seeing any new questions coming in online. So I just wanted to, again, thank everybody for joining us today and being a part of this conversation. And for Brian, thank you for sharing this information with us. I'm assuming that if folks do have questions for you, they can reach out to you directly and you'll be able to get to them and get those answers. So for folks online, if you need more information about something that you were presented today, please let me know and I'll put you in contact with Brian.
Right on. Thank you.
All right. Thanks, everybody. Thanks so much, Brian. We'll see you next time and have a good conversation with our students. Thank you, everybody. We'll see you next week.